guys, welcome back again to my channel. So, last upload or last my video, we'll tackle already about the cellular respiration and fermentation. At kung bago pa sa channel ko, pakipindot naman yung subscribe at notification bell para updated ka sa mga video na ilalabas ko. And now, this video, we will discuss about the photosynthesis or metabolic processes, or how to adapt
our carbon
into the
dioxide
leaf of tree to make an new oxygen.
photosynthesis, the process by which green plants and certain other organisms transform light energy into chemical energy. During photosynthesis in green plants, light energy is captured and used to convert water, carbon dioxide, and mineral into oxygen and energy-rich organic compounds. In order for plants or trees to grow, they need input of carbon dioxide, water, and energy. The chemical process by which plants use this resource to manufacture glucose. The building block of plant is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. Overall reaction of photosynthesis. In chemical term, photosynthesis is a light-energized oxidation-reduction process. Oxidation refers to the removal of electrons from a molecule. Reduction refers to the gain of electrons by a molecule. In a plant, photosynthesis, the energy of light is used to derive the oxidation of water, or H2O, producing oxygen gas or O2, hydrogen ions H+, and electrons. Most of the removed electrons and hydrogen ion ultimately are transferred to carbon dioxide or CO2, which is reduced to organic products. Other electrons and hydrogen ions are used to reduce nitrate and sulfate to amino and sulfidyl group and amino acid, which are the building block of proteins. In most green cells, carbohydrate, especially starch, and the sugar sucrose, are the major direct organic product of photosynthesis. The overall reaction in which carbohydrates, represented by the general formula CH2O, are formed during plant photosynthesis can be indicated by the following equation. This equation is merely a summary statement for the process of photosynthesis. Actually involves numerous reactions catalyzed by enzyme or organic catalysis. These reactions occur in two stages. The light stage, consisting of photochemical or light capturing reaction, and the dark stage, comprising chemical reaction controlled by enzyme. During the first stage, the energy of light is absorbed and used to drive a series of electron transfers, resulting in the synthesis of ATP, and the electron donor reduced nicotine adenine dinucleotide phosphate, or NADPH. During the dark stage, the ATP and NADPH formed in the light capturing reaction are used to reduce carbon dioxide to organic carbon compounds. This assimilation of inorganic carbon into organic compound is called carbon fixation. Light intensity and temperature. As has been mentioned, the complex mechanics of photosynthesis include a photochemical or light harvesting stage in an enzymatic or carbon assimilating stage that involve chemical reaction. This stage can be distinguished by studying the rate of photosynthesis at various degrees of light saturation or intensity and at different temperature. Over a range of moderate temperature and at low to medium light intensity or relative to the normal range of the plant species. The rate of photosynthesis increases as the intensity increases and is relatively independent of temperature. As the light intensity increases to higher level, however, the rate becomes saturated light saturation is achieved at specific light intensity depend on species and growing condition in the light-dependent range before saturation. Therefore, the rate of photosynthesis is determined by the rate of photochemical steps, and a high light intensity some of the chemical reaction of the dark stage become rate limiting. In many land people, a process called photorespiration occur, and its influence upon photosynthesis increased with rising temperature. More specifically, photorespiration competes with photosynthesis and limits further increase in the rate of photosynthesis, especially if the supply of the water is limited. Structural features. The intricate structural organization of the photosynthetic apparatus is essential for the efficient performance of the complex process of photosynthesis. The chloroplast is enclosed in a double outer membrane and its size approximates a spheroid about 2,500 nm thick and 5,000 nm long. Some single-celled algae have one chloroplast that occupy more than half the cell volume. Leaf cells of higher plant contain many chloroplasts, each approximate in size of the one in some algal cell. When thin sections of a chloroplast are examined under the electron microscope, several features are apparent. Chief among these are the intricate internal membrane, or the lamellae, and the stromata, a colorless matrix in which the lamellae are embedded. Also visible are starch granules which appear as dense bodies. The stromata is basically a solution of enzymes and small molecules. The dark reaction occurs in the stromata, the soluble enzyme of which catalyze and conservation of carbon dioxide and mineral to carbohydrate and other organic compounds. The capacity for carbon fixation and reduction is lost if the outer membrane of the chloroplast is broken, allowing the stromata enzyme to leak out. The light reaction occurring exclusively in the thylakoids, the complex structural organization of lamellae is required for proper thylakoids function. Intact thylakoids are necessary for the formation of ATP. Thylakoids that have been broken down to smaller units can no longer form ATP. Even when the conversion of light into chemical energy occurs during electron transport in this unit, such lamellar fragment can carry out the Hell reaction with the transfer of electron from water to NADP+. In diagrams that describe the light reaction stage of photosynthesis, the actual photochemical steps are typically represented by two vertical arrows. These arrows signify that the special pigment P680 and P700 receive light energy from the light harvesting chlorophyll protein molecules and are raised in energy from their ground state to excited state. In their excited state, these pigments are extremely strong reducing agent that quickly transfer electrons to the first acceptor. These first acceptors also are strong reducing agent and rapidly pass electrons to more stable carriers. In light reaction 2, the first acceptor may be pheophytin, which is a molecule similar to chlorophyll that also has a strong reducing potential and quickly transfers electrons to the next acceptor. The special quinones are next in the series. These molecules are similar to plastiquinone. These receive electrons from pheophytin and pass them to the intermediate electron carrier, which include the plastoquinone pole and the cytochromes and B and F associated in a complex with an iron-sulfur protein. In light reaction 1, electrons are passed on to iron-sulfur protein and the lamellar membrane, after which the electrons flow to ferredoxin, a small water-soluble iron-sulfur protein, when NADP+ and a suitable enzyme are present to ferredoxin molecules, carrying one electron each, transfer 2 electrons to NADP+, which pick up a proton or a hydroion and becomes NADPH. This is the Calvin cycle. How the process it?
Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose.
So that's all guys. Thank you for watching.